This video is going to be on mutations, and it's also going to represent the point in the series in which we'll be moving a little bit away from standard general biology and actually into biology as it pertains to evolution. So, as far as mutations go, there are several types, and there's a lot of mystery surrounding mutations as far as the general public is concerned. There are a lot of misconceptions in the sense of that it'll make this horrible creature all the time, that they're always bad and things like that. Those simply aren't the case. What mutations are just changes in the DNA that often arise from copying errors whenever DNA is being transcribed. You can just think about it as if you were to transcribe or write a book by hand, just make a copy of it. Instead of writing "the cat ate the dog," you wrote "the bat ate the dog." That would be an example of a mutation. Now, there are several types of mutations, and I'll start to go into those one by one here in a minute. Also note that this video is going to be covering the common misconception of mutations can't add any new genetic information, as I'll show exactly how that can happen and examples as far as where it has happened. An important thing to understand is that mutations can occur anywhere along the DNA strand. It can happen in the structural portion of a gene, making a completely different three-dimensional protein, and thus having a different function or be functionless. It can also happen in the operator portion of a gene, rendering it always on, only on in the presence of a certain molecule, or always off. It can also occur in non-coding regions. That is, there are certain segments of DNA which don't code for any protein, don't code for any gene, and a mutation can happen there with, relatively little consequence whatsoever. It's also important to note that an organism doesn't know that it needs to mutate. It's not anything that you can control, it simply happens. Also, as much of it happens during the copying of DNA, there are many proofreading mechanisms that DNA goes through, in the sense of after it copies DNA, or after a cell copies DNA when it's beginning to replicate, it'll go through various mismatch repair mechanisms and things like that to ensure that the new copy is correct. And while these safeguards against mutation correct the vast majority of them, every now and then mutations do get through and passed on. It's inevitable. Each of us have plenty of them in our bodies going on every day and every hour. Nevertheless, these mismatch repair mechanisms correct the vast majority of mutations, but they do occur at a set rate, in the sense of every so many generations you will have X mutations passed on to your offspring, or every X cell divisions you will have X number of mutations. Furthermore, we can also take a look at two organisms, related, unrelated, same species or not, and compare the number of mutations that they have, and since they occur at a set rate, we can predict, based on the number of mutations between them, the last common ancestor. This is commonly used, and it's really not up for debate. So, there are also, as I mentioned before, several types of mutations, and I'll start going into those now. A type of mutation called a substitution can occur if you substitute one nitrogenous base for another. So, for example, in the sentence, the cat ate the dog, if you change that to the bat ate the dog, changing the C to the B would be an example of a substitution. Now, this doesn't always lead to a different protein being made, because if you recall the wobble hypothesis, which I showed by this picture earlier in my third video regarding DNA, if you remember, the last codon, or the last base in a codon, can oftentimes be coded for the same amino acid. In other words, you can have AAT and AAC both coding for the same amino acid. So when this happens, there really is no change because you're making the same protein. However, if it does substitute for something else, you can get a completely different amino acid, which can affect the folding and oftentimes structure and function of the finished protein. So another type of mutation can be called just a deletion. It's where you simply have a gene and just a piece of DNA gets lost. It happens most of the time. This is deleterious, however, not always. It just depends on the segment of DNA. For example, if this happens in a non-coding region, it typically won't really hurt anything. Another type of mutation is called an insertion or deletion, and these are called frameshift mutations collectively. If you recall from the third video, DNA has a very distinct reading frame, and that every 3 codons will equal one distinct amino acid. And it goes based on every 3. If you were to insert something or delete a specific base, you would shift the reading frame completely. This results in virtually every amino acid that's coded after that to be completely changed to something completely different. And as such, this will affect the protein folding and the structure and the function. Now, most of the time this induces what's called a stopped codon, and it causes the transcription to stop, so you'll get a short, non-functional protein, but every now and then, in the case of myelinase, you'll get a completely different protein. That is, if the stop codon isn't encountered for many bases along the way. But nevertheless, insertion and deletion mutations are extremely important, and as they allow for the creation of novel enzymes, very rarely, but it absolutely does happen. So if it happens with one, you'll have typically just completely different amino acids following. If it happens in two places, that is, if you have it on position 1 and 2, you'll also get completely different amino acids. But if you have it in 3 times, that is, you have 3 insertions or deletions, that one particular amino acid will be messed up, but everything afterwards will be the same. So think about why this is, because if you remember, it's a 3-base reading frame, so you'll just knock out one portion of the amino acids, and that's very important to understand. The final type of mutation that I'll be going into is called gene duplication. This very commonly happens in that you have a gene and you essentially just duplicate it so that you have an extra copy. So keep in mind, if this gene were to be making a very essential protein for the cell that it needs to live, if it were to be deleted, the cell would die. However, if you were to have an extra copy of it, and something were to happen to one of those extra copies, well, the cell would be fine because it would still be making the original one. And this is what often happens, or it would still have the original intact gene and it'd still be making the original functional protein. So if you go back and actually think about what this means, that means that you have an extra template. You've got this duplicated gene, you've got your other one functioning fine, that you can have all sorts of mutations occurring on in, and basically with no ill effect. This leads to basically kind of your cell can experiment, although it doesn't actually know that that's what it's doing, but it can accrue large differences, all sorts of mutations, and make all sorts of new and interesting proteins, most of which have absolutely no function whatsoever, but occasionally some of them do. And this is exactly how new genetic information is added, by gene duplication followed by subsequent mutation. And nyelinase is an excellent example of this. So to tie a lot of these concepts together, nyelinase, which is a novel enzyme, a brand new enzyme that's was found in bacteria, enabling them to digest nylon. Now we know that this wasn't present in the environment before because nylon has only been around for a couple decades, and since it wouldn't be having any other function, which the nyelinase enzyme doesn't because it's very specific for its substrate, it would have accrued lots of mutations had it been there from the beginning, as there's nothing selecting against them, and remember, mutations occur at a set rate. So we've got this new enzyme. What happened, or what led to its formation, is there was a gene that was duplicated, and from that a frameshift mutation occurred, leading to all sorts of completely different amino acids being formed just from this one mutation. And keep in mind that although the odds of this happening are extremely small, when you have billions of organisms on Earth with very small generation times consistently multiplying over millions of years and billions of years, I mean, this is exactly what happens. These things accumulate, and they're responsible for the amount of diversity that we see today. So again, quick recap. Nyelinase, a brand new enzyme that was formed from the insertion of one amino acid that caused a stop codon 392 positions away. Now, what this did was it allowed for the encoding of all new, completely different, completely random amino acids, which just happened to change the way that the protein would fold. By changing the folding, you're changing its structure. By changing its structure, you're changing its function. So it went from a protein that could not digest enzyme or could not digest nylon to one that completely could, which is exactly how new information is added. And this has always really puzzled me, this argument of no genetic information has ever been added, because it's seen every day. It's nothing new. I mean, we've known about this for years. It's basically in any high school level biology book. I mean, it's fact, it's not up for discussion, and it's always puzzled me exactly why this argument has persisted. I guess, like all things with creationism, it really just kind of stems from misunderstanding. Another example of how you can add new information is basically the addition of any trait. If I were to have any mutation, for example, if I were to have brown hair, and I would have a mutation there, the population of humans would have a mutation that would cause you to have blonde hair, that'd be new information. Even if you are deleting part of the gene that codes for the pigment of hair to one that makes brown pigment to one that makes no pigment or blonde pigment, even if it were a loss of information, it would still be a gain in information because you have an additional type of organism that can be formed. You can either have a brown-haired one or a blonde-haired one, whereas before you could only have brown-haired. This is new information as it's a new trait. Whether or not it comes from the net loss of information or a net gain, it's a complete misunderstanding of how DNA and genetics work in general. This has been a pretty brief introduction to mutations and exactly how they work and what kinds there are. There are several kinds that I know that I didn't list, but they're frankly not relevant to this discussion. Check out the link in the video description as it's to a Wikipedia page regarding mutations, and it's a fairly decent read. It's accurate for